下一个报告人。下一个报告人是来自于美国犹他大学的Dr. Tyler LeBaron， 抱歉啊，他也是从事了很多这个轻生物学的研究，然后呢，他是这个重点在运动生理学、化学的双兼职吧，重点研究心脑血管疾病中氢的这个作用，包括了分子氢、ROS 信号、一氧化氮线粒体运动和关于延缓衰老。他给我们今天带来的题目是 Systemic Therapeutic Effect of Preoperative Molecular Hydrogen Administration. Welcome.
谢谢。Okay, so yeah, this is the perioperative administration of molecular hydrogen. So this is not working. Okay. So the cardiac surgery, including heart transplantation, are linked to multi-organ damage. This is due to a variety of factors, including the ischemia and reperfusion, the use of anesthesia, defibrillation of the cardiac muscle, perioperative hyperoxia, and extracorporeal circulation. A lot of this goes back to the reactive oxygen species, which we understand causes a lot of these problems during the ischemia and reperfusion process. Unfortunately, mainstream antioxidants often do not work, and many studies have shown this. So this gives us the proposition that perhaps we can use molecular hydrogen. We know it has high bioavailability, it has high easy penetration, selective antioxidant effects, enhances endogenous antioxidant system, attenuates excessive inflammation, and favorably influences microRNAs and gene expression. So maybe we should try hydrogen. And in fact, we know that hydrogen has a very long, interesting history. As Professor Lee and others have talked about, we published a timeline history of molecular hydrogen. And going back from billions of years ago, hydrogen was very first. But we found as early as 1793 that hydrogen was reported to have therapeutic effects. So it's a very interesting history. So go to our hypothesis. The perioperative administration of hydrogen gas may mitigate IR, ischemia, reperfusion-induced tissue damage, and oxidative stress to the heart, tissue, kidneys, and various plasma markers. So here's our methods and our biological model. This is a combination of several studies that we did. This is including what Professor Jan Slezak did earlier. So we used these pigs here, 4 to 5 months old, 55, 60 kilograms, and we had a control group and then 2 experimental groups, the transplantation as well as the with transplantation and hydrogen also. And then we did the cardiac surgery. And what we see here is we have the pigs, and we give them the normal anesthesia. We also have the other pig, and this one we're going to do inhalation, as well as incorporate the hydrogen into the extracorporeal circulation. So these are the 2 experimental groups. And then we will collect some of the markers from the plasma and the urine as well. So this is the pig here, and this is doing the research, doing the surgery. It was a very, very interesting time. So go to the results. We saw earlier from the cardiac damage, the heart was actually pretty well taken care of, although hydrogen did appear to have the trend to reduce CK levels and troponin, so had protective effects on the cardiac damage. But what I thought was interesting is there was no change in the NRF2 or KEAP1 and the catalase activity in the left ventricle. And yet, we also see that the H2 attenuated the rise in antioxidant enzymes in the left ventricle. So both the SOD activity SOD1, and catalase activity, if anything, the administration of hydrogen tended to decrease these levels instead of what we normally see, an increase in the antioxidant enzymes. But compare this stands in contrast with the plasma oxidative and antioxidant markers. So in this case, we have the MDA, that was reduced significantly. The 8-hydroxy-2-deoxyguanine was also reduced. Uric acid was reduced, and the GPX activity by hydrogen was also reduced. From one of our first studies, we also saw markers of oxidative stress reduced in the plasma, and markers of MDA and 8-hydroxy-2-deoxyguanosine was also reduced. And then we also looked at in the kidney. So this is the Western blot analysis of the renal cortex, and we found that the NRF2 and KEAP1 levels were higher in the hydrogen-treated group, and also superoxide dismutase levels were also higher in the hydrogen-treated group. So a little bit discrepancy with the left ventricles and the plasma and with other organs. So the question here, H2 treatment only increased antioxidant signaling in plasma and other organs, but not in the left ventricle of the heart. Is this discrepancy paradoxical or instructive? We see some interesting results from previous studies we did. So for example, in this one we have the MDA levels after irradiation. And we see in one of these, after the 45 days, for example, the hydrogen gas concentration on the control led to increased MDA levels. So we sometimes see increased markers of oxidative distress. In fact, in this clinical study that we published relatively recently, when you look at the actual results, we actually saw that there were trends of increased MDA, 8-OHDG, and a decrease in SOD1. And so this is a clinical study administering molecular hydrogen. So this is not so uncommon. Usually we see decreases in oxidative stress, but sometimes we see these mild increases. And I just wanted to point out some of these in the literature. Another study found that the use of inhalation of 2.9% H2 increased the MDA levels in the normal mouse brain by 4-fold. So there's the sham, but then the sham plus hydrogen, MDA levels actually increased. There was a protective effect with hypoxia and the pre-hydrogen, but even after that, with including hydrogen, again we see some increases in MDA levels. So it's a little bit strange, perhaps. However, this is a very fascinating finding as well, where hydrogen increased the mitochondrial potential and the production of superoxide radicals in SY5Y cells. So maybe this could help us understand that hydrogen does have this effect at sometimes increasing superoxide production. And this goes back to this possibility of hormesis, actually. And I've talked about this in several of my articles in the past. H2 may also have beneficial mild pro-oxidant properties by exerting hormetic benefits similar to those produced by exercise. Typically, when we measure the free radicals and oxidative markers after exercise, those are increased. Not a lot compared to smoking or many other like radiation, but they're increased a little bit, followed by benefits. Also wrote H2 acts as a redox adaptogen. Another, H2 transiently increases various biomarkers of cellular stress, followed by their decrease and subsequent improvements in cellular function and clinical parameters. Again, so sometimes it increases, and then later we see it decrease. And then, actually, Dr. Noda mentioned one of our studies where we wrote that, furthermore, H2 may have some hormetic effects, where maybe the benefits of hydrogen mediated by hormetic effects in this case. So, in our conclusion of this, the perioperative administration of H2 protects the heart and other organs. H2 does not always reduce markers of oxidative stress or stimulate antioxidant genes and proteins, even when a clear benefit from H2 is observed, like our NAFLD study, our clinical study, as well as in the heart transplantation. It had beneficial effects, but not all the markers were in the direction we would think. The bidirectional effect of H2 on redox markers may be more instructive than paradoxical. H2 may act as an adaptogenic regulator of redox homeostasis. Results from these preclinical and translational studies warrant future clinical studies on the use of perioperative H2 administration. And with these clinical studies, I wanted to mention some really amazing advancements in hydrogen research in America. I wanted to show this study here that has been approved by the FDA under an IND by Dr. John Keir at Harvard, Boston University. And they're now doing another phase 1 clinical trial called the FAST trial, the safety and efficacy of hydrogen therapy for extracorporeal circulation. So hydrogen is slowly growing in America, and more researchers are doing this, and I'm happy to be a part of a lot of the research going on. So I want to acknowledge a lot of the people who helped with this, and finally, thank you for your attention.
Thank you very much for Dr. LeBaron.